two kind of parts of photosynthesis, photo and synthesis. And photo is the part where light is harvested, and the energy from light is harvested. And synthesis is where we synthesize, we build things, right? So today we're going to focus on the photo, and then on Thursday we'll focus on the sense of the synthesis part. Okay, so we're only doing a few slides today, hopefully give you an opportunity to absorb some of this content, and then we'll wrap up the whole chapter when we talk later in the week. Now, we are going to talk about our light reactions, and in order for us to do that, we probably should review what light is. It's a good start, especially for those of you who haven't taken physics. So, light is actually electromagnetic energy, and we call them waves. But a lot of times when we think about waves at the beach, which is correct, but a lot of times we don't understand what waves actually are. Waves are movements of energy. Waves do not move matter. Okay? Matter can move as a result of the energy that passes through it, but usually when it comes to waves, where the matter ends is the same place as where it started. So I want you to think about if you've ever been to a baseball game and you've done the wave, what do? We do what? You're about to be on video.
I didn't want to be on
Okay.
video.
So you're sitting at the baseball game, and then when it's your turn, you do this. Okay. Do you end where you started? Yeah. Right? So you are the medium of that wave. The energy is moving around, Mile High Stadium or wherever you're at, but you are in the same place at the end as you were at the beginning. That's what a wave does. Okay? A wave is the movement of energy. It moves through a medium. A medium is the matter, but the wave itself doesn't have protons, electrons, or neutrons. Okay? It doesn't have those things. This is important because when we talk about photosynthesis, we're going to capture energy, but we need electrons. Because remember that ultimately our goal is going to be to fix carbon into glucose and reduce it. Well, how do we reduce it? We reduce it by adding electrons, right? But if the energy is coming from the sun, and the energy doesn't have matter, the energy doesn't have electrons. So we have to get the electron from something. Okay, that's where this kind of comes into play. So we need to talk a little bit more about the vocab of waves, the distance between the crests. So a wave, many of them, most of them, look like a sine curve. Okay. And the distance from top to top is what we call the wavelength. Crest to crest is called wavelength. We can spread this out, we can stretch it so that our wavelengths get really far apart, or we can collapse it so our wavelengths are really small. The closer together the crests, the more energy those waves have. Okay. The further away those crests are, the less energy they have. So when we talk about this wavelength, it's actually a length that we can measure in meters. It's a length. How far is it from here to here? And so wavelengths of the electromagnetic spectrum from 380 nanometers to 750 nanometers, that is the visible light. And that's what visible light is. Those are the colors of the rainbow. Roy G. Biv, you may have heard, right? Red, orange, yellow, blue, indigo, violet. Okay? So this is the most important part to today's conversation, but it's not the most important part of your life. Here's the whole electromagnetic spectrum, which is just one kind of wave. There's lots of different kinds of waves, like sound waves and radio waves are here, seismic waves, waves at the beach, these kinds of things. There's waves all over the place. But when I look at the electromagnetic waves, notice our wavelengths. So again, that's going to be our distance from top to top. Waves can be huge. Here's 1 meter, 10 to the third, this is 1,000 meters. 1,000 meters from the top of one wavelength to the next. These are your radio waves. And they are floating around our body right now. Okay, they're all over the place. Are they going to hurt us? Because they're low energy waves. these hurt you? Yeah. Because those are higher energy waves. Okay, these can really hurt you. Hopefully you don't come across a lot of gamma waves. This whole range of waves is actually emitted by our sun. Trick is, most of them are absorbed by our atmosphere before they get to Earth. Okay. Which ones make it through? Visible light and some of these. The fact that some of these get through and they're slightly higher energy than we would like means that we need to do what? Put on sunscreen. Exactly. Higher energy waves are more dangerous. So this energy is going to come back into play here in a little bit when it comes to photons. But look at this. Microwaves. This is how you cook your food. Right? That's an important part of your life, or if it's not yet, it will be when you go to college. Infrared. Most remote controls work off infrared waves. Right? So these waves are a huge part of your life. The ones that matter right now to us are these. Notice how we're pointing out in this diagram the shorter wavelength, the longer wavelength is inverse, longer wavelength, lower energy, shorter wavelength, higher energy. So when we look at a chloroplast, because that's what we're looking at today, that's what we're working with today, when that light, that visible light, meets matter, in this case a leaf, three things can happen. The light could be reflected, the light could be transmitted, or the light could be absorbed. Any of those three. Now, in order to absorb light, only certain substances can do this. Those certain substances are known as pigments. Remember last class I had the test tube with the green stuff in it, and I shined the light on it. From one end it looked green, but on the other end it was like bright red, it looked like blood. That's because there was a pigment in that test tube, and the pigment was absorbing the energy from the light that I was shining on it, but there was nothing for those pigments to give the energy to, because it wasn't in the chloroplast. And so it had to just release that energy. And when it released that energy, it released it through something called fluorescence, which is why it turned red. And then if I could detect tiny changes in temperature, an itty-bitty amount of heat. Like, itty-bitty amount of heat. So pigments, the things that absorb this energy, absorb some wavelengths, but not others. And there's lots of different kinds of pigments, and each different kind of pigment absorbs a particular wavelength. Why? Why does that happen? Well, it happens because within a pigment, here's my pretend pigment atom. A pigment is actually a gigantic molecule, but it has special atoms attached to it. The electrons are very near the nucleus, but when the wave of light, the photon, has an energy level that is exactly the same as the difference in energy levels in the atom. You guys remember learning about energy levels in chemistry? Same thing here. When the photon has the exact amount of energy that is between the energy levels in the atom, that electron bounces up to an excited state. But it's not stable up here, not even sort of stable up here. So it very quickly falls back down. Unless there's something up here to say, hey, hey, I want your energy. Hey, hey, give me your energy, give me your energy. If it just falls back down without giving its energy away, that's when we get the red color and the heat. But if there's another molecule up here that's, I don't know, a protein, that can say, hey, I like that. I'll take that energy. Then we take that energy and we turn it into a high energy molecule. What do we call high energy molecules in the form of ATP? Another high energy molecule, NADPH. Right? So we have to trap the energy when it gets excited and give it to a high energy molecule to then use it to fix carbon and turn it into glucose. Why do we see green when we look at leaves? Because chlorophyll generally absorbs all the colors except green. So all to see is what comes through and bounces off. All the other colors are absorbed. So we call this diagram an absorption spectrum. Okay. The ability of a pigment to absorb different wavelengths of light can be measured with a spectrophotometer. The same kind of thing that we used for that glycol lab that turned brown. So in that case, we started a chemical reaction, then we put it in the spectrophotometer, and our independent variable was time, right? And every, whatever I said, 15 seconds, you took an absorbance measurement. If instead we tweaked the wavelength, so in that experiment you set it to 470 and you didn't touch it, right? You just watched. If our independent variable instead was the wavelength, we could measure what that absorbance value is at different wavelengths. I actually tried to do that with our green stuff from last class. I wonder if I can actually make this. And our spectrophotometers are not sensitive enough. I got either maxed out or I got nothing. Like, all right, fine. I needed better equipment, but I tried. So these are all different pigments. And plants have more than one. So if we look at what wavelengths can be absorbed by a particular plant, it's all of these in the blue and the violet. Not so awesome with the green and the yellow and the orange. But we did pretty good at the red. Right? That's the energies that can be absorbed. And because they're absorbed, we don't see them. It's this part here. We see plants as this green or yellow green. Because that's what's reflected. Yes?
Why are some plants purple then?
They have different pigments. So there's tons and tons and tons of different pigments, and purple is one. It's just not a common one. It's not pictured here. So long before modern day, scientists were trying to figure out a lot about plants. Plants were very confusing and very, very confusing beasts. Scientists thought plants got their mass from the dirt, that they were incorporating mass from the ground that they were planted in, and that's how they got bigger. Because it was kind of a strange thing, right? Like animals eat food, a lot of it, and we get bigger. I'm like, well, how do plants do that? Where do they get their nourishment from? Well, it must be the dirt. So then a scientist actually got a plant, got a bucket of dirt, found the mass of the dirt, found the mass of the plant, put the plant in the dirt, and let it live for 2 years. And in that 2-year period, at the end, he took the plant out of the dirt, found that the plant had gained like 180 pounds, and the dirt had not lost any weight. He's like, something amiss. Well, and it's because the mass of the plant was coming from the CO2. Right? The plant was actually taking CO2 out of the air. That was the first experiment to get scientists to go, okay, we got something to work out here. Right? We didn't understand it. And so we've come a long way since then. But this is an experiment not from that far long ago, but prior to the days of spectrophotometers, this guy named Engelmann was trying to figure out if all light was created equal when it came to this business of photosynthesis. You guys ever seen a prism? You know, people hang them in their windows sometimes, and the light will hit them, and it'll split light into all of its different colors. It'll like shine rainbows around your kitchen. So Engelmann got himself a petri dish, like this, and he put some water in it, and then he put a chunk of algae in it. So algae is something that photosynthesizes, similar to a plant, but different kinds of tissues. And then he got out his fancy prism, and he put it between the light source and the petri dish, so that he got a rainbow in the petri dish right on top of the algae. Then he's like, okay. He put in a whole bunch of aerobic bacteria. And aerobic means that they want what? Oxygen. So then he looked, where do the aerobic bacteria actually go? Well, they went to the place where there is more oxygen. And the more oxygen meant the more photosynthesis. So after doing a lot of experiments, using a lot of different petri dishes and a lot of different poor little bacteria, he was ultimately able to figure out, essentially, an absorption spectra for this particular algae based based on where the bacteria were moving. Pretty ingenious, if you ask me. And if we look at what we have worked with today, if we say, okay, on the whole, where do we see photosynthesis? That is not all that different from what Engelmann picked, what Engelmann developed. So what's so special about pigments? Well, when a molecule of a pigment absorbs that photon of light, one of those electrons jump up, this is what I was saying earlier, to an electron shell where it has more potential energy, and that's when we call it excited. The only photons that are absorbed are those whose energy match exactly the difference in energy from the ground state to the excited state. And that energy is different for different pigments. So a particular pigment is going to absorb photons of light at a particular wavelength, because remember, wavelength matches energy. So pigments are going to absorb at a particular wavelength. Once that pigment's electron has been jumped up to the excited state, it can't stay there for very long because it's so unstable. So the electron drops back down, literally billions of a second. I mean, it's not there for long at all. And excess energy is released as heat, and they give off the photon. It's a glow. We call that fluorescence. That's what I showed you last class. The chlorophyll molecules, when they're in the chloroplast, work with other proteins to harness that energy rather than letting it escape as heat and fluorescence. These proteins are all organized into complexes called photosystems. And a photosystem is composed of what we call a reaction center complex surrounded by several light harvesting complexes. So here's a photosystem. Again, a bunch of proteins. Notice we're in a membrane. So transmembrane protein. Okay, we're in the thylakoid space here, the stroma here. So this is, remember when we talked about the stacks of hamburger buns, this is one of the hamburger buns. Okay, a membrane of that hamburger bun. So what happens is, here's our photon of light. Here's our light coming in, hitting this thing. These green dots are representing our pigment molecules. And that first pigment molecule, it's like setting off a domino reaction, right? The first pigment molecule goes bam, energy, but it can't have energy for long. But there's so many of these pigment molecules jammed in here that when the energy falls, from one molecule of pigment, there's another one right there next to it to grab it. So it's literally like that schoolyard game. You know, we talked about this before, right? We're the frogs, I don't remember the song, right? But we go bounce, 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 bounce. So it bounce, 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 until eventually it ends up here. And this is what we call our reaction center complex. And it's not directed movement. I mean, this kind of seems like it's moving in that direction, and it's not. It's random. Right? The energy goes up, and then as it comes down, it activates another one. The energy goes up. As it comes down, it activates, and so forth. Eventually, they end up here. This right here has huge power. It has the power to split water. Okay? It has the power to split water. And this was actually such a good question that I got this morning that I'm going to put this on mute, and we're going to talk about splitting water. Okay. So we said we were going to split water, and that was going to be the source of what? Why did we need to do this? To get what? Look it up, folks. I'm not telling you. Why are we splitting water? What did water give us? Do you remember? No, not quite. You're close though. What are you going to say? Guess.
No, I was going to say, what did she say?
Oh, she said AC energy. There it is. You got it. Electrons. Perfect. Thank you. We need electrons. Oh, right. We talked about this last class. All right. So if I'm going to say, what does water look like? Right. More or less, that's what water looks like. You guys remember drawing covalent bonds back in the day? That's what water looks like. Excellent. What's the waste from the late reactions? Very good, and it's going to leave in the form of O2, right? Okay, well we have a little bit of a balancing issue here, do we not? Because that's going to take two of these in order to make two of those. Would you agree that that would work? Did I just balance that? Okay, so if that's a water, I need two of them. There's the other one, right? My waste is going to be oxygen here, right? Single bond, double bond. Say it louder. Double bond, right? We have our rule of 8. So that's what we end up with, right? That's O2. How many electrons are present here? 12. Because we have 4 that are shared, right? So we've got 12 electrons. How many electrons do we have here? Well, total, sorry. 8 in each, right? But 16 total. So when we went from here to here, we magically lost 4 electrons. I think that makes me Harry Potter. It's a good day. Are there any electrons there? No, how come not? Positive. So when we call H pluses protons, the reason we call them, because look at an atom of hydrogen. Are there any neutrons in an atom of hydrogen? Not a one, okay? So if it's neutral, it's going to have 1 proton and 1 electron, right? But if we say it's positive, that means it lost its electron. So H plus is just a proton. That's why we call it a proton motive force, right? Because it's just a proton. So are there electrons here? Not a one. So I must be Harry Potter. Unless I'm going to take those electrons and use them to, I don't know, make ATP and reduce an electron carrier such as an ADPH. So when I do this, I have liberated a whole 4 electrons right here. Okay? I need them. Okay, if I'm a plant, I need them a lot. Because remember, what I need to do is reduce my CO2 into glucose. It's all redox reactions. I'm going to reduce my CO2. Look at this again. CO2. What's the difference? Don't count them, but what's the difference between this and this? Hydrogens. It's just hydrogens. So I need to reduce this to turn it into this. How do I reduce stuff? I add electrons. You catch me? So I gotta get the electrons from somewhere. Where am I gonna get the electrons? I'm gonna steal them from water by repurposing them, repurposing the hydrogens and my oxygens as oxygen gas. Okay? So let's go back to this. So, now, okay, so we have our light that comes in, activates an electron, but here's the thing. There isn't an extra electron out here in our pigments. So we have an electron that bounces up an energy level and then immediately falls back down, but it doesn't get to leave the pigment. Okay, so our electrons have to come from somewhere. So we go through this game, right, of, energy up, energy down, energy up, energy down, until we get to here. The special pair of molecules here actually steals the electron, steals them from water. So this pair actually sends up an electron up here. This we call our primary electron acceptor. Now we have a very excitable electron that is primed to do some cellular work for us. We have managed to get an electron and get it excited. So at this point, what we're ready to do, I'll go back to the slide in a second. See, look, this is what we just did. We had water, gave up its electrons, and that's how we got our oxygen. Okay? So we sent it up, we're like, high energy electron! And then we go through an electron transport chain. what happens in an electron transport chain? We gradually fall down in our energy as we go through a series of redox reactions. As we do that, we pump a lot of hydrogen ions against their concentration gradient. Good news. Remember these hydrogens? They went into that place where there's a lot of hydrogen already too. We must not waste our hydrogens. We pumped a bunch of hydrogens across the membrane. The only way those hydrogens can get back where they want is to go through what? The ATP synthase. That same protein that we had in mitochondria is present here in chloroplasts as well. So we produce ATP. Well, now we're halfway home. Here we said the purpose of the light reactions was to trap chemical energy in both ATP and NADPH. Now, when we made the ATP, did we lose the electron? No, it fell down its concentration gradient. But remember, in the electron transport chain in cellular respiration, at the end, the electron made water. Oh, did we notice a trend there? Huh. We started with water this time, we ended with water in cellular respiration, right? So, we still have this electron, but it no longer has as much energy as it once did, because we used it to, create that proton motive force. But that's okay, because we have another light harvesting complex here. We've taken another photon of light. Same thing as before, bounce, 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 bounce. Eventually we get to here, only this one doesn't have to split water because it's getting its electron from the electron transport chain. Shoots the electron up again, but this time that high energy electron goes through an enzyme called NADP plus reductase. Very creative name, isn't it? What do you think it does? I don't know, maybe reduces NADP plus. Okay. And now we make our NADPH. Remember, the goal of the light reactions was to harvest the energy from light and put it into these two molecules. And that's what we've managed to accomplish through an electron transport chain here and a very wisely used enzyme here. We harvested light twice, once here and once here. The photosystems, I'm sorry to tell you, are numbered backwards. 2 and 1. That's the order we discovered them. Meh. They absorb different wavelengths. That makes sense. They're not competing then for the same photons. This one absorbs best at 680 nanometers. This one absorbs best at 700 nanometers. Okay. They're not competing. I'm going to go back and make sure I didn't miss anything. Okay. So, I want to point out some of the similarities between chemiosmosis and chloroplast and mitochondria. Chemiosmosis and the electron transport chain, it's occurring across a membrane in the mitochondria, versus in chloroplast, it's happening again across that thylakoid membrane, but it's always across a membrane. Why is this so important? Because the whole concept works because hydrogen is pumped against its concentration gradient. Well, how do you have a concentration gradient? You have to keep something apart. You have to keep them separated. How do you keep them separated? You use a membrane to do that. Potential energy is stored in the form of that proton gradient, and then the only way those protons can balance themselves out is by going through the ATP synthase. It really is very similar proteins in both of those organelles. There are some key differences. Even though both of these are double membrane, and when we were talking about the mitochondria, that inner membrane was all convoluted, and that's the membrane it happened across. Here, that's not the case. We still have a double membrane, but we're working with these membranes up here in the thylakoids. Okay. This image is color-coded so that the dark area is where you have the higher H plus concentration gradient. The lighter is where we have the lower. And so you can see how this is where we pump our hydrogens here. Okay, we pump our hydrogens here. Okay, but it's still occurring across that membrane. The source of electrons you want to keep straight also. The source of electrons here, of course, was water. The source of electrons here was food. And we eat food for their electrons. We talked about that before. So here's our summary diagram. Light reactions. Photosystem 2 absorbs the light. An electron is stripped from water. Okay. It's excited, goes down the electron transport chain, pumps all kinds of hydrogens in here. The only way the hydrogen can get out is through the ATP synthase. We excite that electron again. It's up here, goes through that fancy protein enzyme so that we can reduce our NADP+. This and this both go to the Calvin cycle. And that's what we're going to talk about next class. So this is the photo part of photosynthesis. Next class we'll talk about the synthesis part of photosynthesis. So at this point, what I'm hopeful for